webinar series and the Phenomics webinar number 8 of our relaunch of this webinar series on phenotyping, which has been established in the course of the COVID-19 pandemic situation and the respective limitations. So this is a format we use here that has made use of pre-recorded videos. We have here some experts being interviewed and having recorded a video, and offering here a live Q&A session for those who are interested, and we'll put that video in the aftermath on our YouTube channels so that further questions come in if needed to be answered. The whole series has been organized by 3 different initiatives. First of them, the International Plant Phenotyping Network. Secondly, the European Infrastructure on Plant Phenotyping called EMPhaSIS. And last but not least, the European project EPPN 2020. So my name being Sven Fahner, I'm based at the Institute of Plant Sciences at Forschungszentrum Jülich, and I'm mainly responsible for the coordination and strategy for the infrastructure. We are very glad that we have another expert here today, Taras Pasternak, who will provide a short summary of his pre-recorded talk shortly. Just some housekeeping rules before passing on to Taras. If there are any questions, please feel free to put them in the chat and we will pick it up. Otherwise, if you are watching this video in the aftermath of that session, then obviously you can use the contacts given by the YouTube channel to bring in your questions and to have it answered by our expert here. So some words on Taras. We are very grateful to have you here, Taras. It's great to have your pre-recorded video seen. Thank you very much for that one. Taras has been working in plant biology since 1983. So he brings in a lot of unique experience towards plant phenotyping. He has been working on multiple projects related to plant phenotyping, stress response, hormonal signaling. His main interest is identifying links between whole plant phenotypes and changes in gene expression and chromatin status of cells. He has published his findings in more than 60 publications so far, which is quite impressive. And you will find some of these publications on the webinar series homepage with some background information. And Taras talked in his pre-recorded video on plant phenotyping from whole plant to sub-cellular resolution. And with that, I would pass over obviously to our expert, because it's his Phenomics webinar role here, and you want to hear him. So Taras, do you want to add some information on your CV maybe? And if so, obviously, we are very happy to see your short summary of the pre-recorded video.
Thank you very much for nice introduction. Here's some short summary from whole plants to single molecule. Phenotyping is the main bottleneck in breeding program. So far, the main focus of phenotyping initially to characterize whole plants or plant organ macroscopically. But this phenotyping cannot help to find the shift between genus and complex traits. Why we need to go to deep phenotyping to see what happens in the cellular level and in the level of chromatin organization, because now we move to the genetic era in which the stress response or development of progress is related genetically, not gene expression. So here today I will present some short summary about deep resolution plant phenotypic platforms, which allow you to go from whole plant level to single cell level to single molecule level with its localization and function. The plant consists from millions of cells, and each cell is different. They have different chromatin status, different gene expression, different function. So, but classical methods of molecular biology extract information from whole cell together, like DNA, like protein isolation from whole plants or from whole organs, which does not give information what happens in the cave cell. That's why aim of our study is to analyze plant organs and whole system with sub-cellular resolution. This contains data for each cell geometry, chromatin organization, cell function within the organ coordinate system. We have 4 options principally: cell geometry, then function, cell division, chromatin status, gene expression, protein complex. So here is examples of phenotyping of the growth plants. Foxtail millet. Here is the whole plants. Here is a plant phenotyping root growths. What are our root numbers, seminal root numbers, and anything else. Here, deep analysis of segmentations and 3D reconstruction. And here you can see, find 20,000 cells of each root, unrolled roots. And here, analysis of cortex 2. Each dots present each cell, and each side of each dot is cell volume. You see how the cell grows in the beginning. And here, the cell volume evolution is metaxillium and vascular tissue in the roots. Here, how we can go from cell phenotyping to whole root phenotyping. Here again, same millet roots, and here you see volume of tissue up to 1 millimeter total volume, and cell number. Volume is in fineness, and cell is a volume, which is numbers. You can see that how much cell volume. It's, for example, 4,200 epidermis cell and 52 metaxillium cell up to 1 millimeter. And here, every cell volume in the root tips. You see that metaxillium have a maximal volume, protoxillium less volume, but other cell is much more smaller. Here's tomato phenotyping. Also, we go from whole root to root tips, double labeling, then cell border and nuclei. And here, then after image processing, with each cohort present each nucleus type, cell type. And here, quantification, we will see then this proliferation zone where cell is divided, produce new cell. Here is a transition zone where it's slightly elongate, and elongation zone where cell rapidly elongate. Not only roots, but shoot phenotyping also possible. Here, arbutus phenotyping, then input images and cell border labeling, segmentation, and mass conversion levels, and native analysis of all cell in the, this is 7,000 cells in these suits after 7 days old. And here then quantitative analysis of each cell in the from these things. And shortly conclusions that this platform allowed to perform multi-channel analysis of cell geometry, cell cycle activity, chromatin organization, protein localization is a cellular resolution. Not any molecular marker required, so then any group species can be used directly without any prior treatment, prior anything else. Why platform can apply for all group species, and all proteins is simpler and can perform quickly, does not require special equipment for scanning except good laser scanning microscope or other type of microscope. And here then the literature, what you can find details of all procedure, all steps, and astrologist to Dr. Paul Pantofon, Thomas Belay, Klaus Palm, Katja and Go and Jeff and Leo Perez. Done. Any question? Any question? Any question coming? Any?
Yes, Tadas, thank you very much. There's some questions around, and I guess I would start from the I would start from the end, talking about the conclusion you have presented, and also talking about the different technologies. So you had, in your talk, you had a kind of impressive amount of methodologies shown that you could use and investigate plants on a molecular level. I think what is interesting for many people, researchers out there, is how would they be able to access these technologies that you have shown? So what is the best way for them if they are interested in using different technologies? What should they do? Should they contact you directly? Are you looking for joint projects, etc.? What is the best way?
The best way, contact me face by face or by mail or by Zoom. And then I can explain principally then there's a list of the literature in the end of the presentation, show you what they need. But principally no equipment need, special equipment need. No any special equipment. Usually need only then some solution for labeling, maybe some antibody, which you can order in the company, commercial company. Then you need proper hand to labeling this. Then you need good microscope, but which is present in every biological center have such a microscope, principally. And then you need only learn how to principally scan images, because not all images can be processing. You need to scan images with certain resolution, certain setting, dynamical correction of the scanning, whatever. And then you need to only to image processing, also need to some practice to training how to best way to processing images. It cannot be explained by protocol because each image is different. You need to see, okay, these images you need to put to this a setting for schematation or the setting for detection. Other images little bit changes, but it's only practice, only, for example, 3-4 times training, and then you can do it yourself. It's not so difficult, principally. But you need to always training and to see hand by hand, face by face, to show that, okay, these images you need to apply it with this protocol for processing, because when you do segmentation, when you do analysis, there's you can show 20 parameters for element size and pixel size, voxel size, whatever, before analysis. You need to show optimal band for your images. You cannot write that, okay, you need to always use 20, 20, 20. Sometimes you need to 25 or 10. Just need to see images, and then when you do it 3-4 times, you can do it optimally with all your images, all your things, whatever.
Yeah, great. Thanks for clarifying that, Taras. You mentioned also that so the multiple opportunities these procedures offer. Two rather small limitations, I would say, or rather one, which is saying that you would have it could be used to most of the crop species, but not to all of them. So maybe you can elaborate a little bit on the limitations that you would see towards crop species.
The principle, no limitation. You will see that, for example, foxtail millet have quite big organs, only as, for example, organ size. Of course, you cannot do analysis 3D with 1 centimeter diameter, for example, stem. But you can principally do up to 1 millimeter. You can do up to 1 millimeter, for example, size. In that other case, you can, for example, make section, manual section, for example, for 1 millimeter size, and then make it analysis of central section of the roots or upper part, down part of the roots is enough, principally, when you see the cylindric roots. You can, for example, section up to 1 millimeter central section. It's enough to go 3D and to get enough signal to be immuno or everything else. It's not the problem, principally. But principally, then leaves is even more easy than the roots, because leaves is usually is not so thin. It's easy to do than the roots. Roots is usually have bigger diameter than the leaves. That's okay. So there's no principal limitation. So we can do principally then all crops possible. Younger, of course, better, but principally you can do even much rare. Only size is some limitation, but you can overcome this microtome section with 1 millimeter section, give you all information of the 3D. You can anyway you have millions of cells in this 1 millimeter section thickness. And no any marker required because there's very important because many people use marker or GFP, RFP, global marker, which require time for crossing, transformation, which can changes the properties of the cell or whiteness, whatever.
Yeah, okay, very good. Thanks for that clarification. Going a little bit more into the experimental setup, you've shown at the beginning of your presentation your approaches towards root phenotyping on other Arabidopsis, and you've also shown this results from volumetric Arabidopsis root phenotyping. I think has been quite early in a pre-recorded talk. So can you give us some information on the experimental design, saying how much plants would you need, how much plants would you need to prepare to get to a kind of a statistically sound
Well, it is 3D principally, then each roots, for example, each root tips have around Arabidopsis have 5,000–6,000 cells up to 1 millimeter. And for millimeter roots up 1 millimeter have around 30,000 cells. So if you do, for example, 5–6 root analysis, it will be enough, I think, to make statistics. But if you always choose roots which is typical for your treatment or for your phenotype, for your whiteness, so avoid them which is not far from, not fit with your average phenotyping. But I mean, not phenotyping cellular, but phenotyping in the whole plant level. Average ones give you the average number principally. Because your analysis always million cell or not, okay. Your analysis in the million, you extracted information from 30,000 cells. Yes, I think that if you have information from 3–4 continuities, so whiteness or dermis whiteness or cortex whiteness, this will be enough to get then principally information about the whole root. But of course, better to have then all 30,000 cell analysis like I showed today. All cell in the 1 grams, 1 Excel file. Volume, for example, position, cell type, and then anything else you can extract. Areas and surface area. Surface area is most important, for example, not root hairs length, but root hairs volume or hair surface important. Root surface important for uncoated soil, for uptake, and also volume of the metaxillium, cortexillium important for transport system. Not dense, but volume. Volume, you see, volume is metaxillium myriad is very big, very large volume to compare with other cells. Maximal cell in the because the function. This is T4 plants require very rapid transport assimilates from shoots and two shoots from the roots.
Yeah. Okay. In many of these experiments you have shown, you did investigate other Arabidopsis plants, I guess, which is a plant heavily used in plant phenotyping. Is there some, I understand, or I guess I understand that this is mainly due to that is a very good plant to use the technologies on. But where do you think there are limitations towards what you find in Arabidopsis to other plants, and how would you evaluate that?
It's not only Arabidopsis. Today I show tomato, for example, then in the paper you can find Medicago also, nodule formation, how it would, for example, the water formation, Medicago, for example. Arabidopsis is a simpler plants model because we can easy extrapolate those data for many other plants, but not so because Arabidopsis is usually unusual plants for many reasons. For example, Arabidopsis have very rapid differentiation, very rapid cell elongation, and then this irreversible differentiation to compare with Medicago or tobacco, for example. But tobacco, you will see publication of tobacco, Medicago, there is a link in my presentation. You find also analysis of tobacco roots, for analysis of tobacco roots with donation, with leaves tobacco, anything else. Principally, there's no limitation. Those small plants, it's a bit easy to do scan, but it's not limiting to Arabidopsis.
Yeah, right. Interesting you raised the tobacco. I remember that you used tobacco for your investigations, tobacco and alfalfa for chromatin organization. So is there some specific reason why you chose these two plants, specifically on chromatin?
Yes, because, for example, then every principal cell have, let's say, half-life to potential, half-life to potential. Principally, every cell in plants through time lost to potential. In Arabidopsis, there is very rapid growth. For example, if you use 1 week old leaves or cotyledon, you never grow any cell division because chromatin is too fast remodeling and too fast then differentiate, means that became irregular, whatever. In tobacco is half-life, let's say 1 month. You can use, for example, then there are no changes in chromatin organization or very little changes in the root or in the leaves in tobacco. That's why it is very important to compare species which is totipotent or not totipotent. Medicago also species which is, you can use natural cortex to induce cell division in protoplast or in nodular. But you can never induce Arabidopsis because Arabidopsis became very irregular, rapid chromatin remodeling, rapid chromatin condensation, and you cannot recover anymore. It's irreversible differentiation. This is the reason why species should be done. One of them which is responsible, responser, and there is no response to compare the reason of response.
Yeah. Okay, thanks for that one. Taking a little different angle on the on the technologies and methods that you apply. Now, looking from a breeding angle and from a practical plant breeding angle, where do you think that this would create some benefits in breeding programs or breeding approaches as it is usually done by now, as we know it?
A breeding program now is widely reproduced, widely CRISPR editing plants, for example. And you need to know then how your gene is affected, for example, phenotype. If you want to, for example, have plants with more yield, with more than spike number or more stem number, you need to see how it changes, for example, early events. When you see, for example, plants with more spike or with this, you need to see what happens, more elongation, more which stage they induce. Induced, for example, usually spike inducing the small stem cell in the shoots. And how they induce principally. If you know this, you see early events. You see maybe more targetedly, for example, hey, maybe you can use clavata or vox or boost genes to see more stem in the bead or with or other things. So important to see early events, because when you make full phenotype, you see later events. then you see per request and you see the reason why editing, for example, of the bush or other genus can change these things. And you see idea which protein can interact with this. And then you might maybe not only the genus, but interacting partner also. Because one genus, okay, they may affect, but they're also interaction partner important. If you have one genus changes, interaction partner is no changes, you will have little effect. If you have two interaction partner changes, you will have maybe more strong effect on breeding program. This is just hypothetically, but principally this need to be more investigate, more whatever.
Right. Yes. I'm just looking and encouraging, so from the audience we have, if there's some questions that you would like to raise, so this is your chance now to put it in the chat. If there's something that you would like to bring forward to Taras. Otherwise, we have covered all the input and questions that we have so far. So saying that, I'm just going to leave a couple of seconds and see if there's some questions from the actual audience. Otherwise, I would like to mention also that obviously once this video is available, then for all of those that are watching it, then please feel free to come up with your questions also in the aftermath or when you watch it and bring it forward, so that you get your points clarified. So there doesn't seem to be any.
One more thing. Principally, people have some question after video or after them watching this, they can directly ask me by email or by other way of communication to see then what is then really they want to do and how I can help, how I can set methods in open background, whatever, in what really need which microscope, which labeling, and detail, because this is need to always not theory practice. Need to always have some practical training to get them methods working. You cannot describe it in the like you drive the car. You cannot explain how to drive the car, because you need to always make your open hand. You feel your images, feel your labeling, feel your image analysis. Then need to at least 1, 2, 3, 4 times repeat with every step, adjust images to some practice. This is important. The people always can contact me directly, and then I'm happy to answer, to help, to do setting these protocols in places.
Right, that's very good to know, Taras. Thanks for that. I guess our audience will really appreciate that, and obviously these are kind of great approaches and methodologies, and this would be really. I'm really looking forward to get some further contacts established and some approaches towards your side to get it used and to get it made use of. Taras, do you have any further points that you would raise at that moment? Otherwise, I would start closing the session informally, but I would just kind of open the floor for you if there's something that you would like to bring up.
Principally, yes, I always happy, open to cooperation, open to work, open to discussion. That people can directly contact me anytime. Then we can discuss the ability to common work or to anything else, to help to setting methods, to help to protocol or whatever. This is what I want to tell to people who potentially will look this video in the YouTube or in the internet, whatever.
Right. Very well received. Thanks for that, Taras.
Thank you.
Obviously, also thanks a lot for the video that you've made, which is very impressive, very informative. Thanks for your time you spent with us today. And yeah, looking forward to see about the feedback that you receive or that we receive based on this video now. And as you said, or to all the people watching the video, feel free to contact also Taras directly. He's very open and very much looking forward to your feedback and to you getting in touch with him. Right, unfortunately, this is then kind of the end of that session, which is also the end of this current Phenomics webinar series. But we are already in preparation of the next series. So please stay informed, stay tuned via the newsletter that you could use, also via the homepage. And I guess we are very much looking forward to have similar presentations like from Taras in the next webinar series, which is a great experience and which is great input, obviously, to the community. So thanks again, Taras. Thanks to everybody.
Thank you.
And also thanks to all the people that will still watch the video in the future.
Okay, thank you. Bye.